welcome to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And this is for the chapter Biomolecules. And we are in part 1 of the series. Alright, so let's begin. Okay, so question number 1. Identify the basic amino acid from the following. Okay, this appeared in NEET 2020, pretty recent. A. Tyrosine, B. Glutamic acid, C. Lysine, and D. Valine. Okay, so or valine, whichever way you pronounce it. So you find the basic amino acid from the following, and the correct answer is C. Lysine. Okay, so answer is C. Let's go to the discussion. Okay, so basic amino acids are what? Dibasic and monocarboxylic acids. Okay, so they have a dibasic, they have two NH2 groups and one carboxylic group. Okay, that's how you identify a basic amino acid. Okay, this is a very good important point to remember. Okay, if any structure is given to you, if you see dibasic and monocarboxylic acid, then you know that it is a basic amino acid. Okay, so they are highly basic in nature. There are three basic amino acids, that's lysine, arginine, and histidine. Okay, so the structure over here that you see is that of lysine. Okay, you have this NH3 plus group here, NH3 plus group here, and then you have one carboxylic group here. So now you can easily know that it is a dibasic monocarboxylic acid, which is basic in nature. Sorry about that, which is basic in nature. All right. Okay, let's move on then. This was pretty direct, and I believe in NCERT the example lysine is mentioned there, but you should ideally know how to identify a basic amino acid. Okay, and these are the three amino acids that are basic in nature, so you can study them. All right, let's move on then. Question number 2. The two functional groups characteristic of sugars are, and this appeared in NEET 2018, A. Hydroxyl and methyl, no. Carbonyl and methyl, no. Carbonyl and phosphate, no. Carbonyl and hydroxyl, yes. Okay, so these are the two functional groups that are found in sugars. Okay, in carbohydrates. Answer is D. All right, let's go to the discussion. Carbohydrates contain carbonyl group, that's C double bond O group, and hydroxyl group. You can see the OH group over here. So these are two groups, functional groups, that are very distinct for carbohydrates. Okay, and it is easy to identify that's a carbohydrate. So remember, it's a carbonyl group and a hydroxyl group, so on methyl or phosphate or any of the other groups that are mentioned in the options. All right. Oh yeah, so this is it. This is the answer. I guess we've answered the question. Two functional groups characteristic of sugar, yeah, carbonyl and hydroxyl. Right, let's move on. Till now we've got very direct, easy question. Question number 3. A typical fat molecule is made up of, and this appeared in NEET 1 2016, A. 1 glycerol and 1 fatty acid molecule, 3 glycerol and 3 fatty acid molecules, C. 3 glycerol molecules and 1 fatty acid molecule, D. 1 glycerol and 3 fatty acid molecules. So typical fat molecule, basically they're talking about triglycerides. Okay, it's a typical fat molecule which is found in our body also. Okay, when fats break down, they form triglycerides. Okay, so the answer is D. It has 1 glycerol molecule and 3 fatty acid molecules. We'll look at the structure. So the answer here is D. If it's a monoglyceride or a diglyceride, then it will be different. They'll have 1 fatty acid or 2 fatty acid. But here we are specifically talking about triglycerides as a typical fat molecule. That was the question. So a typical fat molecule is made up of 1 glycerol molecule and 3 fatty acids. That's triglyceride. Okay, so this is a general structure that's taken from NCERT. So you should be aware of this, of a triglyceride. So you have 1 glycerol molecule and there is esterified with fatty acids, that's R1, R2 and R3. These are variable groups, so it can be saturated fatty acids, it could be unsaturated fatty acids, whatever the case is, in R1, then in R2 and then in R3. R3. Okay. So R1, R2, and R3 are the fatty acids. All right, let's move on then. Question number 4. A phosphoglyceride is always made up of, and this appeared in NEET 2013. A saturated or unsaturated fatty acid esterified to a glycerol molecule to which a phosphate group is also attached. It's pretty much that. Saturated or unsaturated fatty acid esterified to a phosphate group which is also attached to a glycerol molecule. Not really. Only a saturated fatty acid esterified to a glycerol molecule to which a phosphate group is also attached. Only an unsaturated fatty acid esterified to a glycerol molecule to which a phosphate group is also attached. Alright, so very long options given here. So we're basically talking about phosphoglyceride. So phosphoglyceride can have a saturated or unsaturated fatty acid esterified to a glycerol molecule, which we just saw in triglycerides, to which a phosphate group is also attached. It's the only reason why it says phosphoglyceride. Alright, so the answer is A. Okay, so the correct answer is A. Let's go to the discussion. So the something's wrong with the pointer here. Glycerophospholipids or phosphoglycerides. So if they write phosphoglycerides, you know what they're talking about. They're talking about if they write glycerophospholipids, also you know what they're talking about. Consists of glycerol 3 phosphate. That means the phosphate is attached to the third carbon atom. To the third carbon atom. Okay. And esterified at C1 and C2 with fatty acids. So C1, this is C2, esterified with fatty acid like we saw in triglycerides. Okay, CH, the third carbon atom, that is, has the phosphate group. Okay, has a phosphate group. Now what you see here, the structure, you see the choline group also attached to the phosphate group. This is actually a typical structure of lecithin. Okay, lecithin is an example of a phosphoglyceride. Okay, it is a type of phosphoglyceride. So if they even mention lecithin, I think it's then one of the questions. If they ever write lecithin, they're talking about phosphoglyceride. So this is what you see here is a typical structure of lecithin. Okay, which is a type of phosphoglyceride. So lecithin has this normal phosphoglyceride structure. Along with that, it has a choline group attached to it. Okay. All right, so I guess I've answered the question. If we look at option A, saturated or unsaturated fatty acid esterified to a glycerol molecule to which a phosphate group is also attached. All right, let's move on. Okay, so question number 5. Uridine present only in RNA is a Sapien Karnataka NEET 2013. A nucleoside, B nucleotide, C purine, and D pyrimidine. So uridine we're talking about is a nucleoside. Okay, so the answer is A. So uridine is present in RNA and is a nucleoside which contains base and sugar. So there are if you're talking about RNA, then the base is actually uracil, which is a pyrimidine. So if they had written uracil, it is a nitrogenous base, which is a pyrimidine. All right. Now they've spoken about uridine. What they have mentioned in the question is uridine. So uridine is present in RNA, of course. We know that, which consists of, it's a combination of base and sugar. That means uracil base plus the pentose sugar together forms nucleoside. Now if uracil, if this was the formula given to you, plus pentose sugar, plus the phosphate group attached to it, then what will happen? That means it's a nucleotide. Okay, it's a nucleotide. Okay, so it's uracil plus pentose sugar plus the phosphate group attached together forms a nucleotide. Uracil plus pentose sugar forms nucleoside. Uracil itself is a nitrogenous base, also known as the pyrimidine. Okay, and yeah, that's the answer. So again, this is important. You need to remember what is the difference between uridine and uracil. Don't get confused. Very easy to then select pyrimidine and then you unnecessarily get the question wrong. So it's a nucleoside. Okay, yeah. Move on to question number 6. Which one out of A to D given below correctly represents the structural formula of the basic amino acid? Let me see. Yep. Okay, sorry. All right, so we have to look for basic amino acid, and we are in this appeared in the year 2012. So you have A, B, C, and D. Now I already spoke about basic amino acid. Remember in the first question, it's a dibasic monocarboxylic acid. Okay. Let's see. Let's look for dibasic. Is A dibasic? No. Is B dibasic? No. C dibasic? No. D. You have NH2 here, you have NH2 here, you have COOH here. So D is a basic amino acid. So the option over here is B. Please don't select option D. Okay, it's a very common way of confusing yourself. So you look at the option D and the D is right, so you select the option D. No, the answer is D, but your option is B. All right. So the correct answer here is B. Okay, so structural formula in D is that of lysine, which is a basic amino acid. We already looked at lysine in the first question, right? And already discussed what basic amino acids are and how to identify them. Right, let's move on. Question number 7. The given diagrammatic representation shows one of the categories of small molecular weight organic compounds in the living tissues. Identify the category shown and the one blank component X in it. All right, it's appeared in 2012. So you have a pentose sugar here, which looks like a ribose sugar. It's not deoxyribose, but oxygen molecules are present there, right? So it's not a deoxyribose sugar. So you have category A cholesterol. No, it can't be cholesterol. Amino acid. No, definitely not. C, it's a nucleotide. No, it's not a nucleotide. D, nucleoside. Yes, because nucleotide, it can't be a nucleotide, there's no phosphoric acid attached to it, right? It only has a pentose sugar plus a base, which is X over here. Okay, so it definitely has to be nucleoside and uracil. It's a pentose sugar. It's not a that means a ribose sugar. It's not a deoxyribose sugar. Okay, deoxyribose means the oxygen atom is not present. Okay, it's absent. So nucleoside, and yes, the base is uracil. So the answer is D. Okay, so the X component has to be a base. This is how nucleoside is structured. Okay, it has a base and it has a sugar, pentose sugar attached to it. So that's a nucleoside, which is uracil. Okay, so let's go to the discussion. So structure is that of a ribose sugar present in RNA, and the X component is uracil, which is present in RNA, forming a nucleoside. All right, so you already discussed nucleoside earlier. So we'll move on now. Question number 8. Which one of the following biomolecules is correctly characterized? It's appeared in mains 2012. A. Lecithin, a phosphorylated glyceride found in cell membrane. B. Palmitic acid, an unsaturated fatty acid with 18 carbon atoms. C. Adenylate acid, adenosine with a glucose phosphate molecule. D. Alanine amino acid contains an amino group and an acidic group anywhere in the molecule. Okay, so yes, lecithin, a phosphorylated glyceride found in cell membrane. That is the right answer. Answer is A. Let's go to the discussion. Okay, so lecithin is a glycerophospholipid found in cell membrane. Already discussed lecithin. It has a choline group attached to the phosphate molecule and is found in the cell membrane. Okay, palmitic acid is a saturated fatty acid and contains 16 carbon atoms. All right, adenylate acid is adenine with ribose phosphate molecule. Okay, adenine is the base with the ribose phosphate molecule. Alanine amino group contains an amino group and a carboxylate acid group attached to the carbon atom. As you can see here, this is a structure of a alanine amino acid. So you have a carbon atom over here to which NH2 and carboxylic acid is attached to it. All right. Okay, let's move on then. Question number 9. Which one of the following structural formulae of two organic compounds is correctly identified along with its related function? This appeared in 2011. So you have A and you have B. Option A, B, adenine, a nucleotide that makes up nucleic acids. No, it's not a nucleotide that makes up nucleic acid. B, A, triglyceride, major source of energy. Okay, B, uracil, a component of DNA. And A, lecithin, a component of cell membrane. Yes. So the correct option is D, A, lecithin, a component of cell membrane. It is not, of course, the structure over here is that of adenine, but it is not a nucleotide that makes up the nucleic acids. Alright, so the answer over here is D. And over here I've already discussed lecithin. So lecithin seems to be a very important structure to remember. So please remember this if they ever ask you in the exam. Structure A is lecithin, which is a component of cell membrane. Structure B is adenine, which is a nitrogenous base called purine. Okay. So yeah, this was simple. You can move on then. Question number 10. About 98% of the mass of every living organism is composed of just 6 elements, including carbon, hydrogen, nitrogen, oxygen, and this appeared in 2007. So 98% of mass is composed of just 6 elements. Okay, just matter 98%. And what are these? Carbon, hydrogen, nitrogen, oxygen, and sulfur and magnesium? No. Magnesium, sodium? No. Calcium and phosphorus? No. It's phosphorus and sulfur. Okay. Answer is D. Please remember this fact. Okay, there are 6 elements that are found in this mass of every living organism, and these are the 6 elements. All right. The 6 elements that are present in the living organism are listed over here. Please remember them as it is. Okay, if they ever ask you, just remember this as a list, note it down in your notebook for you to study later on. All right. So it's pretty direct, but it's easy to confuse yourself sometimes. You get confused whether it's in sodium or calcium. No, but it's phosphorus and sulfur. We know carbon, hydrogen, oxygen, nitrogen are the major 6 elements. Of course, apart from that, there are major 4 elements, but apart from that there are 2 others, phosphorus and sulfur. All right, let's move on. Question number 11. Which of the following is the simplest amino acid? This appeared in 2005. A, alanine, B, asparagine, C, glycine, D, tyrosine. Simplest amino acid is C, glycine. Answer is C. So glycine is the simplest amino acid present. It does not contain any R or variable group. Remember, when we look at the other amino acids, there are various other variable groups attached to it, right? But this has no any R or any variable groups attached to it. It is simple. This is the structure of glycine. It only has one hydrogen atom here. All right. And you have a carboxylic group here, you have the NH amino group over here, carboxylic group over here, and you have 2 hydrogen. All right, so it's a very basic structure. All right, let's move on. All right, so question number 12. The major role of minor elements inside living organism is to act as, this appeared in 2003. A, cofactors of enzymes. B, building blocks of important amino acids. C, constituent of hormones. D, binder of cell structure. So the major role of minor elements, okay, the way it's phrased is funny. But yeah, we're talking about minor elements and what they majorly do. So according to the options given over here, they are cofactors of enzymes. Okay, the cofactors of enzymes. The answer is A. Let's go to the discussion. Now we know primarily that enzymes are proteins, right? So an enzyme not only contains the protein part, but there is a non-protein part also to enzymes. Okay, they are known as cofactors, which behaves like a catalyst in reactions. Okay, so they enhance the rate of reaction. The cofactors can either be organic or inorganic in nature. Yeah, the metal ions are inorganic, right? Metal ions are inorganic, like calcium, magnesium, molybdenum. These are some of the inorganic cofactors that are there. They're also known as activator, okay, which attach to the active sites of enzymes and enhance enzyme activity. Okay, now coenzymes are non-protein organic factors that are loosely bound to the enzyme. Okay, we spoke about cofactors, which are either organic, and that's a very important concept to understand in enzymes. We'll also come to enzymes later on, but cofactors are either organic or inorganic. Now let's go to the inorganic part. The inorganic part are those metal ions that we spoke about. Okay, they're the minor elements. Okay, and they are known as activators, which enhance the rate of the reaction. Now, there's another non-protein organic factor which are known as coenzymes that are loosely bound to the protein part. Okay, they are known as coenzymes. So, the non-protein organic cofactors are that are loosely bound are called as cofactors. Coenzymes, I'm sorry. A prosthetic group is non-protein organic cofactor that is tightly bound to the enzyme. So, the one that is loosely bound cofactor is called coenzyme, and prosthetic group is the cofactor which is tightly bound to the protein part of the enzyme. Okay, so like I said, cofactor, that's one definition that you need to remember. Coenzyme, prosthetic group. Okay, so these three terms, the definition of these three terms you need to remember. Okay, also, like I said, the metal ions are known as activators. So the inorganic part, that is the inorganic part of the cofactor. Okay, you need to remember these three terms. And for your sake, please note it down in your notebooks, study it whenever you need to. But yeah, these are the four definitions that you really need to understand when it comes to enzymes. Okay, when you're studying the structure of enzymes. Okay, so as I said, cofactor can be either organic or inorganic. Inorganic part are the metal ions, which are known as activators. Then you have the organic part, which are loosely bound. They are known as coenzymes, which are tightly bound, are known as prosthetic group. Simple to understand. Okay, read it over and over again to remember. Okay, so if we go back to our question, the major role of minor elements inside living organism is to act as cofactors of enzyme. Can't be building blocks of important amino acids. No, that's different. Constituent of hormones, no. And binder of cell structure, no. So this is the major role. Okay, let's move on then. Question number 13. Lipids are insoluble in water because lipid molecules are, somewhere in 2002, hydrophilic, hydrophobic, neutrals, bitter ions. So they are insoluble in water because lipid molecules are hydrophobic. It's basically the definition of hydrophobic molecule. They are insoluble in water. They are afraid of water basically. Hydrophobia is like when you're afraid of water. So lipids are generally afraid of water. Yeah, that's their nature. Okay, that's how they are formed. So the answer here is B. Let's go to the discussion. So yeah, lipids are insoluble in water because they are non-polar compounds. Okay, they are non-polar compounds. So the hydrocarbon chain, which is predominantly present in lipids, makes the lipid non-polar. So you can see here, this is the structure of a typical lipid molecule. Okay, so you have a hydrophobic hydrocarbon chain. This hydrocarbon chain is what makes the lipid non-polar. Okay, and water is polar molecule, so they don't mix together. You can see, you can even experiment this at home. Take a drop of oil and put it on water. They're not going to mix. You'll see the oil floating in water. And you have this hydrophilic carboxyl group, which is the head, which loves water. Okay, so the hydrophilic part will be soluble in water, but the hydrophobic part, that is the hydrocarbon chain, will not be soluble in water. But the majority part of the lipid is hydrocarbon, this chain over here, the hydrophobic part. The majority part of the structure of a lipid is majorly hydrocarbon, which is non-polar. There's a very small part which is polar, which makes it soluble in water, but that really doesn't make a difference. Majorly, it is insoluble in water. That's what it is. Okay, so which a hydrocarbon chain which is predominantly present in lipids, and this makes it non-polar. Since water is a polar solvent lipid, will not be able to dissolve in it, hence lipids are hydrophobic in nature. So like I said, water is polar, and only the polar part will then dissolve, and that's a very small part of the whole lipid chain, right? So the hydrophobic part forms a major part of the lipid chain. So yeah, they are majorly insoluble in water and they are hydrophobic in nature. All right. It's a good question. I mean, very direct when it came to answering the question, but it was important to understand this information. How are lipids structurally formed? Why is it hydrophobic in nature? So you need to remember this. Okay, let's move on. Question number 14. Spoilage of oil can be detected by which fatty acids? Appeared in 2001. A. Oleic acid, B. Linolenic acid, C. Linoleic acid, and D. Erucic acid. Okay, so spoilage of oil can be detected by D. Erucic acid. Okay, so the answer is D. So erucic acid is a monounsaturated omega-9 fatty acid and is easily degraded by microorganisms, which produces that foul taste, okay, and smell. Learn this as a fact. Learn this as a fact. Okay, that erucic acid can be used to detect the spoilage of oil. Okay, and yeah, so this was pretty like it was direct. The information isn't really available on NCRT, but I would suggest you just study it as a fact. Okay. So question number 15. Essential amino acid is, so we are in 2000. A. Phenylalanine, B. Glycine, C. Aspartic acid, and D. Serine. So essential amino acid is A. Phenylalanine. Okay, answer is A. So yeah, amino acids, essential amino acids are amino acids that cannot be synthesized by the body and need to be supplied through the diet. That's why they're essential. They're basically essential to us. Okay, that's how they are termed essential amino acids. Okay, they cannot be synthesized by the body. And yeah, this is a list of the essential amino acids, out of which phenylalanine is one of the options. I would suggest you study this list, note it down. If they ever ask you, what are the essential amino acids, they give you like 4 options and they ask you to choose from it. Yep, this is pretty much that. Let's go to the question. Yep, glycine, aspartic acid, and serine. No, they're none in the list. It's only phenylalanine. Okay, this was a direct. Like I said, note this list down and remember it. Okay, let's move on. Question number 16. What are the most diverse molecules in the cell? This appeared in 1996. A. Lipids, B. Mineral salts, C. Proteins, D. Carbohydrates. The most diverse molecules in the cell are C. Proteins. Answer is C. So the most diverse molecules present in cell is proteins. Each cell contains 1,000 different types of proteins and they perform varied functions. Yes, of course, like they play a very important role in forming the cell structure. Then hormones are enzymes are also proteins. Then you have antibodies, which are proteins. Yep, so these are some of the functions. They're varied functions, they're diverse. Now we know that there are 20 amino acids present, right, in our body that are present in nature. And so proteins are polymers of these 20 amino acids. Okay, so the different combination, permutation, combination of those 20 amino acids which are forming these proteins, right? And these proteins then in turn, play a role in forming the cell structure, hormones, enzymes, their antibodies, and so on. So yes, proteins are the most diverse in cells. Okay, the most diverse molecules in cells, because of the 20 amino acids, the different permutation combinations, out of which so many different types of proteins are being formed, and these proteins then in turn, take part in different activities in the body, like I've mentioned a few. Muscles are there which are formed of proteins, and so on. So they are basically, they actually the building blocks of life. Proteins, when they say, when you read in your textbooks that they're building blocks of life, yes, that's true, because without proteins our bodies wouldn't exist like that. We can't just survive on carbohydrates and fats. We need proteins too. Okay. And yeah, this is it. Okay. So what are the most diverse molecules? Remember this, it's proteins. All right, let's move on. Question number 17. The four elements that make up 99% of all elements found in a living system are, this appear in 94, we already discussed this before. A, CHO and P, CNO and P, CHO and N, CHO and S. So it's C, hydrogen, oxygen, carbon and nitrogen. Answer is C. Okay, so it's carbon, hydrogen, oxygen, nitrogen. Okay, so this is a table. So you see hydrogen on top, you have carbon, you have oxygen, you have nitrogen, you have sulfur here. Unfortunately they haven't given the percentage of phosphorus, but also yeah, these are the actually 6 elements that are found, but they have already over here mentioned 4 elements. So yeah, this is taken from NCRT, by the way, so you should know this table. All right, let's move on then. Question number 18. Amino acids are mostly synthesized from, and this appeared in 1992, A. Mineral salts, B. Fatty acids, C. Volatile acids, and D. Alpha ketoglutarate acid. So, amino acids are mostly synthesized from alpha ketoglutarate acid. Answer is D. So, yeah, let's go to the discussion. So, most amino acids are formed from alpha keto acids and then undergo transamination, usually from glutamate, to form new amino acids. Okay, so alpha keto acid is the precursor, okay, that then undergoes transformation from glutamate. That means it's a transfer of amino group, okay, to form again new amino acids. Okay, and the reaction is such that alpha keto acids plus glutamate, it's a reversible reaction, forms amino acid plus alpha ketoglutarate. Okay, so alpha keto acid is first that forms an amino acid and then undergoes transamination, usually from glutamate, to form again new amino acids. So the reaction also forms alpha ketoglutarate, which forms glutamate by the process of amination. So alpha ketoglutarate is used to again then form glutamate, to again then which undergoes amination, okay, by the process of amination. So alpha ketoglutarate also plays a role in forming the amino acids. It's a little complicated, but I would suggest don't spend too much time on this biochemistry part. But just understand that alpha ketoglutarate plays a role in forming that glutamate, which then by the process of amination, which then again goes under transamination to form new amino acids. Okay, so most amino acids are formed this way. Now some amino acids such as glutamate, glutamine, proline and arginine are synthesized by alpha ketoglutarate. I mean, they just need alpha ketoglutarate to form, like we've seen previously. See how alpha ketoglutarate is used to form glutamine, glutamate, proline and arginine. Okay, so these amino acids belong to the alpha ketoglutarate family. Okay, so like in the option it said alpha ketoglutarate Yeah, at alpha ketoglutarate acid. So yeah, alpha ketoglutarate acid. So yeah, that is the appropriate option amongst A, B, C, and D here. But like I said, just understand, it's superficial, and I agree it's superficial, but we'll not spend too much time on the biochemistry part of the amino acid synthesis for now. Okay. But I just wanted to show you how alpha ketoglutarate plays a role. Yes, there are some amino acids where alpha ketoglutarate is used directly. Then there's another reaction where alpha ketoglutarate is used to form glutamate, which then undergoes transamination to form new amino acids along with alpha keto acids. So this is the reaction that's basically happening. All right, let's move on then. Question number 19. Living cell contains 60 to 95% water. Water present in human body is, this appeared in 1992, A 60 to 65%, B 50 to 55%, C 75 to 80%, D 65 to 70%. So living cell contains 60 to 95% water. Water present in human bodies, that means the whole body, how much water is present there? Answer is A 60 to 65%. Answer is A. So the water present in the human body for man weighing 65 kg, let's say there's a normal man which weighs around 65 kg to 70 kg, a range. Some textbooks say 70 kg, some say 65 kg, and it is more than 60% of the weight. That means it will vary from 60 to 65%. Okay, now there's a close call between 60 to 65 and 65 to 70%, which is not correct. Medical textbooks say it's 60 to 65%, basically around 60%. You can see here chemical composition of a normal man, weight 65 kg. So water over here has a percentage of 61.6, okay, which is in this range and contributes 40 kgs of the total weight of the body. Okay, so it's more than half. Yeah, so just remember it's 60 to 65% and not 65 to 70%. All right. For your sake, I have referenced Guyton, which is a textbook of medical physiology. Don't worry, you'll come across this when you study MBBS. And there's another book called Biochemistry by Dr. Satyanarayan. Okay. So these are two textbooks, and they have both mentioned 60 to 65%. I would suggest you remember this table, okay, so it'll be useful. You find the chemical composition of all the macronutrients that are there, micro and macronutrients that are there in our body. What is the composition, how much do they contribute to the weight of the body? All right, let's move on then. Question number 20. Secondary metabolites such as nicotine, strychnine, and caffeine are produced by plants for their, this appeared in NEET 2020, pretty recent. A, nutritive value, B, growth response, C, defense action, and D, effect on reproduction. So yes, the products that I mentioned here are secondary metabolites, and they are produced by plants for their, if you have to choose the appropriate answer, it would be defense action. Okay, so the answer is C. Now I have mentioned the role of secondary metabolites. Oh, I'm sorry. Mentioned the role of secondary metabolites here. There are four roles, functions. So they attract animals for pollination, seed dispersal. They're used by plants in their defense against herbivores and pathogens. They act as agents of plant-to-plant competition. They're used in making drugs, insecticides, flavors, pigments, scents, rubber, spices, and industrial materials like gums, resins for human welfare. So these are the basic roles. Although there's some textbooks that say that these are some of the roles that are established now, but we are currently, this is a topic that's undergoing research also. So these are some of the roles that are already established and of secondary metabolites, and yeah, you can study them. Note this down in your notebooks, so you can study them for the exam. All right, let's move on. Question number 21. Concanavalent A is, this appeared in NEET 2019, it's a pigment, an alkaloid, an essential oil, a D-alectin. So concanavalent A is D-alectin. Okay, so the answer is D. So concanavalent A is a lectin and a secondary metabolite. So it is a naturally occurring protein found in most plants. So this is a table taken from NCRT, where they have given a list of secondary metabolites and the examples. So you can clearly see here lectin, concanavalent A, pretty obvious, right? Yep, this is it then. All right, can move on, I guess. I think you answered the question. Okay, so we come to the end of this video lecture, and I hope you've enjoyed today's video lecture. I hope you've learned something new today. And please don't forget to like, share, and subscribe to MJ NEET Biology. And I'm available on Facebook, Twitter, and Instagram, so you can follow me there for more updates. And I'll see you in the next video lecture. Right? So bye.